Hello, my name is Joseph Schmalz, and I am a graduate student at the University of Idaho. And welcome to my presentation titled Spatiotemporal Dopaminergic Modulation of Schaffer Collateral-CA1 Plasticity, a Computational Modeling Approach. I will begin with an overview of the different types of computational models in the neuroscience community, then talk about the challenges of understanding how multiple neurochemicals present in stress affect learning and memory. Next, I will move into the existing experimental studies, then how we developed our computational framework, and then go through how our model compares with the experimental results. And finally, some takeaways from our approach. Since the 1950s, electrical models of the membrane potential of single neurons have existed. Typically, these models capture the membrane potential of the cell body of the neuron. Similar to a real neuron, when enough excitatory input from the dendrites enters the cell body, an action potential occurs, which is a rapid depolarization of the membrane potential followed by a rapid repolarization, as you can see in the plots on the right. When an action potential occurs, the depolarization travels down the axon. Here, at the end of the axon, the spiking neuron sends information to another neuron through the release of neurotransmitters. As you can see in the plots on the right, with multiple action potentials, this occurs very quickly, is on the order of milliseconds. There also exists much slower biochemical dynamics that govern the activity of the neuron, especially at the synapse, which is the connection between two neurons. Here I am showing you an example of one of the biochemical pathways present in the striatum of the basal ganglia. This biochemical pathway is activated by dopamine and acetylcholine binding to their corresponding receptors. As you can see here, these dynamics involve multiple reactions and intermediates, which can be hard to observe in experiment. Furthermore, as you see in the plots on the right of one of the biochemical proteins, the timescale is much slower and on the order of minutes, not milliseconds, which we saw in the previous slide. Because of the timescale difference between the biochemical and electrical dynamics, typically researchers focus on only one or the other. This simplification overlooks any important crosstalk that may occur between these two types of dynamics. So here we want to develop a unified model that integrates both biochemical and electrical dynamics together so that we can understand how brain neurochemicals released during stress impact learning and memory. Stress-mediated disorders such as post-traumatic stress disorder and major depressive disorder are estimated to cost the United States approximately $100 billion in lost productivity each year. These stress-mediated disorders are caused by the release of multiple neurochemicals in the brain. Additionally, different levels of stress will release different levels of each neurochemical and at different times. Therefore, different levels of stress will have a different impact on important aspects of our life, such as learning and memory. As we are all aware, a little stress in our life can have a positive effect, such as helping us study for a test or meet an upcoming deadline. On the other hand, prolonged stress can produce negative effects, like PTSD, which can impair our memory. Therefore, the neuroscience community is interested in studying the effect of each of these neurochemicals on learning and memory. Historically, changes in the strength of the Schaffer Collateral-CA1 pyramidal synapse, highlighted here in the hippocampus, has been used as a model for studying learning and memory. Many studies have been conducted on the effect of each one of these neurochemicals and also on the electrical stimulation at different frequencies of the Schaffer collateral axon on synaptic plasticity at the Schaffer Collateral-CA1 pyramidal synapse. Therefore, experimentally, we have a very clear understanding how each individual neurochemical affects the Schaffer Collateral-CA1 synaptic plasticity, but we do not have a very strong understanding of how multiple neurochemicals interact, or how neurochemicals interact with electrical stimulation at different timings to modulate the synaptic plasticity. In particular, we don't have a clear understanding of how the release of the neurochemical dopamine at different timings relative to electrical stimulation of the Schaffer collateral axon at various frequencies modulates Schaffer Collateral-CA1 pyramidal synaptic plasticity. To fill in this gap, we develop a biophysical model of the Schaffer Collateral-CA1 pyramidal synapse in order to understand the spatiotemporal dopaminergic modulation of this synapse. In order to study Schaffer collateral synaptic plasticity, we must first have a way to measure it. Experimentally, this is done by inserting a stimulating electrode into the Schaffer collateral axon, as shown here on the left, then placing an extracellular recording electrode in the CA1 region of the hippocampus so that the field potentials of the CA1 pyramidal neurons are recorded. Then a small electrical pulse is applied to the Schaffer collateral axon through the stimulating electrode. This induces a small field excitatory postsynaptic potential, abbreviated as FEPSP, that is measured by the recording electrode. Small electrical pulses are continually applied at regular intervals throughout the experiment that this recording electrode then measures. An example of this recording is shown in the figure on the top right. Then the slopes of each of these FEPSPs is measured and normalized to the first one. This gives you the graph shown on the bottom right, where prior to high-frequency stimulation of the Schaffer collateral axon, there is no change from the baseline. But after high-frequency stimulation, the slopes of the FEPSPs increase due to an increase in the synaptic strength. Based on this existing FEPSP experimental data, we develop a computational framework to describe the biochemical and electrical dynamics involved in spatiotemporal dopaminergic modulation of Schaffer Collateral-CA1 pyramidal synaptic plasticity. Our model has two inputs. The first is the electrical stimulation, which depending on the frequency can either increase or decrease the synaptic strength. The second is a dopamine agonist, which on its own slowly potentiates the synapse. Additionally, these two signals interact spatiotemporally to alter the synaptic strength. We developed this model with the following framework. We took the limited experimental data and formed a model hypothesis. Then we checked to see if this model is able to qualitatively describe the experimental data. If it does not, we revise our model hypothesis until it is able to qualitatively capture the experimental data. We then take our qualitative model and fit it so that our model will now make quantitative predictions. We fit it with an approximate Bayesian computation sequential Monte Carlo method that gives us the distribution of model parameters that explains the data. The basics of this approach are as follows. First, we choose a prior distribution that we sample model parameters from until our model output matches the experimental data to within some tolerance. If the sampled model parameters produce an output that falls outside of the tolerance, then the parameters are discarded and new parameters are sampled. If the output falls within the tolerance, then the parameters are accepted. We repeat this until we have a large number of accepted parameters, giving us the posterior distribution. For the sequential Monte Carlo variation, we then repeat this process, only now this posterior distribution becomes our new prior distribution. We repeat this until we have an acceptable tolerance level. Now I will show how well our model is able to capture the dynamics of the dopaminergic modulation of the Schaffer Collateral-CA1 synaptic plasticity. As I mentioned earlier, the frequency of the electrical stimulation will dictate if the electrical input will increase or decrease the synaptic strength. Here, high-frequency stimulation is applied to potentiate the synapse. In the first figure A, the dopaminergic D1-D5 receptor agonist, SKF, is applied more than 200 minutes prior to the high-frequency stimulation protocol. Before the high-frequency stimulation protocol is applied, there is a slow potentiation that is induced by the dopaminergic agonist, shown as the red circles here, which our model is able to capture, shown as the blue line. Then, when the high-frequency stimulation protocol is applied, the synapse is potentiated further and then slowly decays back to the base case of high-frequency stimulation only, which our model is able to capture as well. Next, in B, the reverse order is considered, where high-frequency stimulation is applied first, followed by the dopaminergic agonist. Experimentally, it was found that the that the dopaminergic agonist was not able to further potentiate the synapse after the high-frequency stimulation protocol. Our model was able to capture this time dependency and lack of potentiation as well. In C, similar to A, another experimentalist released dopamine 160 minutes before a high-frequency stimulation protocol. Again, dopamine caused a slow potentiation, which our model captured. Then, when the high-frequency stimulation protocol was applied, the synapse was again potentiated further. This added potentiation again slowly decayed back to the pure high-frequency stimulation case, which was observed in our model as well. Next, we examine how well our model captures the dopaminergic modulation of low-frequency stimulation of the Schaffer Collateral-CA1 synapse. As I mentioned before, the frequency of the electrical stimulation will dictate if the electrical input will increase or decrease the synaptic strength. Here, low-frequency stimulation is applied, which depresses the synaptic strength. The long-term depression induced by the low-frequency stimulation protocol can be seen in these two figures as the black square lines. Experimentally, it was shown that the application of the dopaminergic agonist SKF right after the low-frequency stimulation protocol led to the complete reversal of low-frequency stimulation-induced long-term depression. We applied the same protocol to our model. As you can see in the blue line in A, the application of the dopaminergic agonist SKF after low-frequency stimulation-produced protocol reversed the low-frequency stimulation-induced long-term depression in our model as well. Next, it was shown in an experiment that the application of the dopaminergic agonist 1 hour after the low-frequency stimulation protocol produced only a slight potentiation that was not significant. Again, our model also produced a very small potentiation when SKF was released 1 hour after the low-frequency stimulation protocol. This shows our model is able to capture the time-dependent dopaminergic modulation of the Schaffer Collateral-CA1 synaptic plasticity. Next, we consider other low-frequency stimulation protocols. Previously, we considered a low-frequency stimulation protocol of 1200 pulses at 3 Hz. In A, we consider 2 rounds of this low-frequency stimulation protocol with a 5-minute rest period between the 2 protocols. Then, after the second low-frequency stimulation protocol, the dopaminergic agonist SKF is applied for 20 minutes, which produces approximately a 15% potentiation over the pure low-frequency stimulation case. Our model captures the final synaptic strength measured in the experimental data, shown as the red circles, fairly well, but slightly overpredicts the initial transient synaptic strength change right after the low-frequency stimulation protocol. Next, we then consider the low-frequency stimulation protocol of 2400 pulses at 3 Hz, followed by the application of the dopaminergic agonist SKF. Again, the application of the dopaminergic agonist produces a small potentiation of the synaptic strength after the low-frequency stimulation protocol, but again, the application of SKF doesn't completely reverse the low-frequency stimulation-induced long-term depression. Again, our model is able to capture the reduced potentiation. This further supports that our model is able to handle a diverse set of low-frequency stimulation protocols in combination with dopaminergic agonists. Next, we consider the application of a low concentration of the dopaminergic agonist SKF during a low-frequency stimulation protocol. Experimentally, it was shown that the application of SKF during a low-frequency stimulation protocol enhanced the low-frequency stimulation-induced long-term depression by approximately 20%. We applied the same protocol to our model, and which also produced an enhancement of the low-frequency stimulation-induced long-term depression by approximately 20% as well. This highlights our model is able to handle different concentrations of dopaminergic agonist in combination with low-frequency stimulation protocols. In conclusion, we have developed a biochemically inspired dynamical model that is able to quantitatively describe the spatiotemporal dopaminergic modulation of the Schaffer Collateral-CA1 pyramidal synaptic strength. Our model is able to handle a diverse range of high-frequency and low-frequency stimulation protocols. Also, the model is capable of handling different concentrations of dopaminergic agonists at different timings. The model will now be used to extend the experimental data and fill in the gaps of our understanding. We then plan to experimentally validate the quantitative model predictions. Thank you, and I would be happy to answer any questions.